Hello friends, welcome to BMH Learning. This lecture is about actin polymerization. In the last video, we discussed different cytoskeletal filaments. If you haven't gone through the introductory portion of the cytoskeleton, in the I button you can find the link. So this tutorial will deal with how the actin subunits are assembled to form actin filaments. In the cell, the actin is found in two forms. First is the G-actin, the monomeric globular form of actin. Now, the G-actin monomers polymerize to form actin filaments, and this filamentous form is known as F-actin. The G-actin is generally bound to ATP nucleotide. The ATP binding to the G-actin has a profound effect on the actin polymerization. We will exploit this property as the lecture proceeds. Now, the process of actin polymerization. This process is comprised of three steps: nucleation, elongation, and then steady state. The nucleation is initiated when two actin monomers bind to each other and form a dimer. This complex is weak and can easily dissociate unless a third monomer adds to form a more stable trimer. The trimers are also referred as nuclei because they initiate and accelerate the process of monomer assembly. Now comes the elongation phase. The short oligomer rapidly increases in length by the addition of actin monomers. Let's know something important here. The actin filament which is growing has got two ends, the plus end and the minus end. The actin can be added either to the plus end or to the minus side of actin filament, but it's the case when we have high concentration of free actin monomers. Of course, other cases we will discuss very shortly. Now keep in mind, plus end is fast growing while the minus end is slow growing. So having said that, the rate of addition is nearly 10 times faster at the plus end than at the minus end. Okay? Also, the polymerization rate on the both ends is dependent on concentration of free actin. Now, as the filament piles up more actin units and grows in length, the number of free actin monomers decreases, and it goes on decreasing until an equilibrium is established between the filament ends and the free actin monomers. To make this concept more understandable, we can say the concentration of free actin keep on decreasing until it reaches a constant value, and at this constant concentration value, the rate of monomer addition equals the rate of monomer loss. So, wouldn't you like to know what this constant value is named as? It's called as critical concentration. And this non-varying state is what we refer as steady state phase. So, in steady state phase, if one monomer is added at the plus end, at the same time, one monomer is removed from the minus side, like this. That means no change in the total length of the filament. So, critical concentration says a lot about filament growth. Below critical concentration, filament will not form. Above critical concentration, filaments will form because more free actin is present and the length of filament increases. And finally, at steady state, equilibrium is observed. Now, regarding why do we need this actin polymerization process, let me take you to the main function of actin filaments. And what's that? It's simply the cell movement. So let's discuss how the actin polymerization is involved in cell migration. Suppose we have this actin filament. The minus end and the plus end of this filament are placed at points A and B respectively. Now imagine the filament has to migrate to the point C. Okay, so think the actin polymerization is in steady state phase. That means if we are adding one actin subunit at plus end, simultaneously one actin subunit would be removed, right? And you can notice while doing this, the filament has progressed a bit towards point C. So if we continue this process of addition and removal of actin subunits, our filament will ultimately reach to the point C. So this is how individual pieces of actin are shuffled to move the entire cell around in an environment. Now it's very important. This process of moving the cell by actin polymerization and depolymerization is called as actin treadmilling. Now there arises another question. Who is powering the actin filaments to treadmill? It's ATP hydrolysis. Actually, the free actin are bound to ATP, and actin with ATP are added on the plus end. And then after actin ATP hydrolysis, these monomers are released as actin ADP at the minus end. The free ADP actin is subsequently reloaded with ATP and can participate in the process of actin polymerization. This was all. Thanks for watching.